webinar. It's 7 p.m. Our speakers are here for you this evening. You are now more than 18 participants to this webinar. I will give directly the floor to Daniela Salvatori. Please, Daniela, go ahead.
Yes, good evening or good morning to everybody according to which part of the world you are. Also on behalf of Catherine from CAT and from Nick that is joining me this evening from Interniche and me from Ombion, we welcome to this webinar. Before introducing the speakers of this evening, I would like to give the word to Nick just to announce the next webinar, the date, the speaker, and the topic. Nick, are you okay to intervene?
Thank you. Yes. The next webinar is in about 1 month, specifically on Wednesday, February the 18th. And this is at an earlier time, 10 a.m. Central European time. And this is the session being organised by Catherine Herman from the Centre for Alternatives to Animal Testing, CAT. And in this session, Brett Lidbury, who is an associate professor from the National Centre for Epidemiology and Population Health, NCEPH, at the Australian National University in Canberra, will reflect on his scientific transition from animal-based disease models to human data-driven in silico approaches, and what this shift means for education, training, and the future of fundamental biomedical research. So the link for this will be posted at the end of this webinar in the chat when I remind everyone again of what's coming up next. But for now, back to this today's session.
Thank you, Nick. This evening we have 3 speakers from Utrecht University and specifically from the veterinary faculty. And I am also working with these 3 researchers. So I am very proud to announce their webinar. They will talk about the AvatarZoo. But what is the AvatarZoo? The AvatarZoo started with the need to introduce a new technology in education, a new technology that has a direct influence in also decreasing the number of laboratory animals used in education. Our education also in life sciences, when we talk about anatomy, clinical procedure, tends to be quite classical. The introduction of this new technology asks for a lot of changes and a lot of work from starting understanding how to produce the model, how to present the model to the student, and how also to evaluate the possibility to learn with these technologies in the best possible way. So Berend Heerke, Noemi Passarello, and Bart van Rossum will give you a bit of a flavor of what we are doing at Utrecht University at the vet faculty. They will also briefly present themselves with their expertise before starting their presentation. So I would like to give the word to them, and who will be the first to start? You need to tell me this, Berend. Berend Heerke is the technology innovator for the veterinary faculty, and he is also responsible for the skills lab. So Berend, the floor is yours.
Thank you very much, Daniela. I will try to share my screen and see if the right version of the presentation is shared. Thank you very much for the invitation and a warm welcome to everybody here in this meeting. We are going to talk about, as Daniela mentioned, the AvatarZoo, reducing laboratory animal use or animal use in general, in education through XR. And I realized just a minute ago that I perhaps need to explain the term XR and digital plastination. Bart's going to tell everything about digital plastination, so please bear with us until he is presenting. XR stands for extended reality, and extended reality is the full spectrum of virtual reality, augmented reality, and mixed reality. And these are all different technologies which are now kind of combined in the term extended reality, and I will show you some use cases of that. As Daniela mentioned, today Noemi, Bart, and myself will present something about the AvatarZoo, the development of the AvatarZoo, and the research we are doing with the AvatarZoo. So AvatarZoo is a very cool initiative, a cool program, although I realize that it's not so cool anymore to say cool, but anyways. It's a very nice program we are developing at the moment, essentially to learn anatomy in a digital environment. We started out with developing for the HoloLens, and the HoloLens, you see me on the left side of the screen wearing the HoloLens. It's a Microsoft device. It's a device for augmented reality, and that means you can see through the glasses, and the glasses project new information inside your own environment. And as I said, I will show you a short video how that looks like. Unfortunately, last year Microsoft decided not to take HoloLens development any further, so they terminate the HoloLens program. So at this moment, we are in the transition to a more virtual reality approach. And as soon as that is in place, we will inform you through our website. As Daniela mentioned, teaching animals, experimental animals, are often used in education, both for basic science, basic anatomy, and also for training clinical procedures. On the right side, you can see basic anatomy in a dissection setting, and we are very fortunate in the Netherlands to have this donation program fostered by the Proefdiervrij Foundation that is facilitating animal owners, to donate their animal to the vet faculty, to the vet school, when their animal is deceased. So we are very fortunate to have this very ethical source of animals to and to be able to teach our students basic anatomy based on these animals. For clinical procedures, it's a little bit more difficult because you often need live animals for that. But we are exploring the use of AvatarZoo as a simulation platform, and I'm going to shortly tell you something about that also. So first, I will show you a video so you have an impression what the AvatarZoo is like. So you are looking through the glasses, through the HoloLens in this case, and you can see the very nice educational environment that we have in Utrecht University, and you can see an added digital specimen. In this case, it's a dog. It's the German Shepherd. And you can also see a little bit of a user interface, a menu, and this model is fully interactive. You can grab stuff with this beam. You can also grab it with your hands. And as soon as you grab it, the name of this structure is depicted in the text box above. So you can add features that you would normally not have in an anatomy situation. And this animal is fully interactive and fully 3D. So you can see depth, you can walk around it, you can even walk through it, and you can pick up, for example, muscles, take them off and see what is underneath them. This is what we call virtual dissection. So it looks very nice. It's very interactive and intuitive, and the students like to work with it very much. So we're also exploring the use of the AvatarZoo for clinical procedures. In this case, oh, uh, uh, it is as you can see, a cow, and the students need to learn drawing blood from a cow, and they do that from the neck vein or from the tail. And we can animate these animals to make them useful for this clinical procedure. We can strip layers off, so we can make it a little bit easier. We can even strip off the muscles so they can see what they are doing when they're starting to learn the procedure. And for this procedure, for example, they need to lift the tail and they can use the animated model to do so. We are in the first steps of exploring these features as the AvatarZoo working as a true medical simulator for veterinary education. So besides the more obvious features of such a digital digital environment, a learning environment. It's available 24/7. It's very safe, of course. You can make mistakes. You don't harm anybody, not yourself, and also not the animal when you're doing something wrong. You can enrich the learning experience by, for example, by providing the names of the structures that you are working with. But you can also add features like measuring competence, how competent students are in a simulator session, in a similar procedure. And the advantage of this this technology, this XR technology with wearing the goggles, is that you essentially have a computer on your head that can measure a lot of things and show you the digital content in 3D. And the technology behind that is based on our own vision. You may know that we are seeing depth because the image in our left and our right eye is a little bit shifted and a little bit rotated. And that's why we see in 3D. And seeing things in a three-dimensional aspect is very important. Gathering spatial knowledge. The other thing that's very important is allowing people to move themselves around a subject. That's what we call dynamic exploration. And that's also helping to build up this spatial recognition and spatial knowledge in your brain. And this spatial knowledge is very important because essentially we only realized for a couple of years now that this is the way that we have learned anatomy, and we are still teaching anatomy nowadays. A lot with books, flat media like cell phones or tablets, and even with 3D objects like this human lower leg. But these 3D objects are presented on a flat screen. And that's a little bit of a difficulty because this flat information, and Noemi is going to tell you a lot more about that, but I will just kick off a little bit. This flat information, as you learned it from a flat media, is stored in your brain as flat information. But in clinical practice, there's hardly any any patient that is 2D, right? That's flat. So every medical person, medical doctor, veterinary doctor, needs to convert this flat information into spatial information, applicable spatial information. And that's not so obvious. You need something that is called spatial ability and more focused, the ability to mentally rotate flat images into the 3D space. And we can measure how good people are with that. This is a very old psychology test from 1978. I was 10 years old then. And essentially the test works like this. On the left hand side, you see an image of a 3D object. In this case, it's a figure with 10 little cubes in a special orientation. And then you need to decide which of the 4 images on the right side are rotated images of the left image. In other words, which of the images on the right side can you rotate to get the image on the left side? It's 2 images, you can try it. And the other 2 images are usually mirror images, and you cannot rotate them to the desired picture on the left side. So I give you a little bit of time to decide that it's the first and the third image that are rotatable into the left-hand image, and the other 2 are not. So this very simple test gives you a score, and when you look at the scores in the population, these are usually normally distributed. So most people score around the mean here, and about half of the people score above, and about half of the people have a score below the mean. And we have done a lot of research in the recent past, to look at different projection technologies. So 2D from a book, or 2D from a screen, something we call a 2.5D. 2.5D is showing a 3D image on a flat computer screen, just like the human lower leg that I showed you before. And 3D is real 3D, real 3D, or digital 3D with wearing the goggles. And then when you see that if you are so fortunate that your score on this mental rotation test is above average, then essentially you can cope with every type of modality. On the other hand, when you are unfortunate and your score is below average, then you are very bad in translating this 2.5D information into spatial information. You have problems with that because you cannot, well, essentially we don't know why. And Noemi is going to tell you a little bit more about that. So there's a good thing and there's a bad thing. About half of the students do very good anyway. They don't need the goggles. They don't need the AvatarZoo, although it's very fun to use it, and there are all these other advantages. But the other half of our student population cannot really cope with this 2D flat information and translate that mentally into 3-dimensional information. We have about 2,000 students walking around at the Utrecht University VET faculty. So about 1,000 of those students have problems converting 2D into spatial information. So that's a lot of people. So I think it's essential to start thinking about how we can help these students. We can help them by letting them learn in this 3D learning environment. Of course, it's not the only way of providing spatial knowledge or 3D knowledge to the students. You can use a dissection setting, as you can see on the right-hand side. On the back, you can see, by the way, a horse that is plastinated. So it's a real horse that is plastinated. All the fat and water molecules are exchanged with plastics. And Bart is going to tell you everything about this technique and what we can do in the context of AvatarZoo with this plastinated specimen. But going back to 3D learning, of course, there's 3D learning in the curriculum. Although the allocated time for 3D learning, for these kinds of dissection courses is getting less and less. It's expensive, the logistics are a nightmare, and the scalability is awful. And of course, we are using animals for that, experimental animals or ethically sourced animals. Anyway, animals for that. Um, so AvatarZoo is, with this introduction, I introduce you to AvatarZoo, our 3D learning environment for learning anatomy and clinical procedures. When you scan the QR code on the right side, you will find yourself on our website. It's a platform where you that provides interactive learning, so you can interact with the models. We have all these models. We are working on to make all these models available. It's experiential learning, so learning by experience, by really pulling out stuff from the animal, looking at it from all sides, and seeing what's behind these structures. It's embodied. You use your own body to learn, to gather knowledge, and that's a very strong learning feature. And we are working on collaborative learning. So we are working on making this environment shareable, so you can two or more people can work on the same model. So with that, I would like to give you the opportunity to ask one or two questions, and then we can proceed to Bart's presentation and Noemi's presentation. Bart will show you a little bit about the plastinated specimen, and Noemi will tell you much more about the research we are doing. And afterwards, if there's time, we are all there to further answer any questions. So thank you for your attention, and if you have a short question, then I'm very willing to answer that.
Thank you, Beren. So you have heard Beren. If you have any question, feel free to share it in the chat, and I will communicate it to Beren. Otherwise, if you have no question for the moment, you can keep it, and we also give time at the end of the webinar. So we'll wait 1 minute or two to see if someone shares something in the chat, and after we can move to the next session. Yes, please, Daniela.
Yes, thank you, Berend, for sharing the work. I am curious if there are colleagues in the audience that are using virtual reality in their education.
So if there is colleagues, feel free to share it in the chat. We have a question from Nick Beren. Will you be sharing these models freely with the world or use them in-house only?
That's a very good question. We are not sharing them at the moment because we're still working on finalizing and reviewing the anatomy of our models. We will be sharing them on a VR platform, but since we have to transition from the Microsoft HoloLens to the VR platform, that will take a little bit of time. We hope that later this year we will be able to share them. We will probably not be able to share them freely for the whole world because it takes a lot of money to make these models and to keep the whole digital environment in place. So we will probably be asking a small fee, but we will do our best to make it a small fee.
Thanks, Beren. We have one additional question, and after we can move to the next session. So how do you manage to let the student stop the blood flow without a resistance if you touch the cow neck to draw blood, for example?
Yeah, that's also a very good question. Thank you for that. Of course, with this technology, by just using this technology, you don't have any haptic feedback. You don't feel anything. So that is of concern, and that is why we are investigating the use of these models in a simulator session. Yeah, in a simulator setting, I should say. So we're not sure how essential that is, and of course, feeling the pulse of the blood, knowing whether you are hitting an artery or a vein, for example, that can be quite essential. Um, uh, we do think, however, that we can teach the students to get a stepwise approach to this clinical procedure and to familiarize themselves with that. And in the end, we can also add haptic features or maybe even add haptic gloves to the setting to provide them with some haptic feedback. So we are fully prepared for that. Only the technology, especially technology for haptic feedback gloves, is at the moment it's not good enough for our training purposes. But as soon as it is, then we will be the first to introduce that into our platform.
Yeah, thanks, Beren, for the answer to Naomi. We can move now to the next session. Please feel free to share the screen.
All right. Well, thanks so much. My name is Bart van Rossum. I am a veterinarian and a research assistant for the Anatomy and Physiology department at Utrecht University. And I've been working on the AvatarZoo project for close to or over a year now at this point. And I will be talking to you about plastination and digitization in anatomy teaching and learning and why we need it. Well, as Berend highlighted, students can learn anatomy from a very wide array of sources. So we have the two-dimensional sources and we have the three-dimensional sources. And for 3D, we usually look at dissections, for example, but that costs cadavers, which is, well, it requires animals, and this is, well, it raises animal concern, or it raises ethical concerns. It costs money, it costs time. It's not very sustainable. So, and on the other hand, we also know that the anatomy curricula are, anatomy is being allocated less time in medical curricula. So we're trying to find basically a solution to offer students the same amount of interactions with spatial anatomy without requiring more animals, or even while requiring less animals. That would be optimal, of course. So something that could offer a solution there is plastination. And as Berend said, in plastination, basically the moisture and fat tissue in animal products are replaced with a curable polymer or plastic. And with that, the biological material is kind of safeguarded and it's protected against the wear and tear that would normally happen to physical specimens. So it can be used multiple times. It can be used over a long period of time because it doesn't really perish like biological materials do. And they can serve as anatomical references, but also as training tools for clinical procedures. But, and that's the big but, they're still based on animal materials, and the specimens can be fragile and availability can be limited. So, for example, at Utrecht University, we have a very large collection of these plastinated specimens, and that can be used inside and outside of the classroom. But this is not the case everywhere, of course. And even in our case, the availability is still limited because students can't really take the plastinated specimens home. So in order to reduce the amount of cadavers needed while increasing their availability, this research project kind of focused on digitizing two plastinated specimens by using accessible methods. So the specimens were turned into high quality interactive digital 3D models, and afterwards we did a small scale evaluation of their usability. And we also wrote an article on the procedure of digitization, which was published recently. So I will show a QR code later if you're interested in reading it. So let's look at the digital model creation. So first, of course, we need to understand what makes up a digital model. So we have the shape of a model, which is called the geometry, and we have textures, which are kind of the images that are overlaid over the geometry. And together they make one digital model. And how can you make them? Well, there's a wide array of possibilities. And we selected 2 different specimens to kind of see what the extent was, or the extent of the possibilities was of our chosen method. So we have a plastinated specimen of the hind leg of the dog on the left and of the knee of a horse on the right. And the hind leg of the dog mainly features a lot of muscles and a lot of muscle fiber details. So therefore, high quality textures would be required to properly digitize it. And the horse knee mostly has like bones and deep ligaments and tendons, so high quality geometry would be preferred. So we started by using a 3D surface scanner, which is basically a combination of cameras and light projectors, which you move around an object. And by doing so, you generate a digital 3D model. And the nice part about these scanners is that they generate a very high quality geometry, but the textures may lack in resolution. The images overlaid over the model can be very blurry if they're generated at all. So this was also the case for us. So what we also did is we used a photogrammetry style image overlay. And with photogrammetry, which is the image on the right here, you basically make a lot of photographs around an object, and you upload all those photographs into software, and the software generates a digital model for you. And this, because it uses the photographs that you've made with your camera, the textures can be very high quality, but the geometry, again, can lack. So we have the surface scanners, which create very high quality geometry, and we have the photogrammetry, which creates very high quality textures. And because they kind of complement each other, we decided to use a hybrid approach, so combine the 2. And well, that's exactly what we did. And that results in digital 3D models that have, well, high quality textures and geometry. So then we used 3D editing software to isolate and annotate all the structures that are visible in the model, so all the muscles and all the ligaments and all the bones, and uploaded them onto our online viewer and also in the extended reality glasses that we have for our AvatarZoo project. And then we take a look at the results. So we started with scanning, which again creates a geometry, which you can see here. And then we have the textures from the scanner. And when we zoom in a little bit, we can see that the textures are a little bit blurry. You can see where the structures are, of course, but that's mainly due to the geometry, and the textures of the muscles and all the different structures inside is, yeah, it's not great. So then we have our image series, which is just a compilation of images from all around an object, and we overlay that, and that's how we get our high quality models. And then the next step was, of course, the editing in 3D software and the annotation function, and uploading onto our online viewer, and that's when we get these results. So you can see that we can see the model, and we can select different objects that are inside. And we also did our small scale evaluation, which was very positive, and a lot of possibilities were envisioned by the participants, but a controlled study will still be needed to get some like proper and clear results. So when we look at the result of the model, you can see it here in this video. You can zoom, you can rotate, and you can select the structures that are inside the models. So the creation was very successful, you can say, but what can we do with these models? Because, well, it would be nice to apply them somewhere. So the high level of detail that we can see in the models, they enable the easy identification of fine anatomical structures. You can really see where all the muscles start and end, especially when you use the annotation function. So because of this, students can use the 3D models as a digital tutor, kind of. They can answer their own questions when they're inside of a classroom. They can be used alongside the physical plastinated specimens, and because they can open the models on any device they want, basically. So on a smartphone, on a tablet, on a laptop, or on the head-mounted displays, the HoloLenses. And outside of the classroom, the models can be used as well, because the students can use them before class as preparation. They can use them after class. as a review, or they can use them as self-assessment because of the annotation function again. So our approach not only enhances learning, but it also provides students with a more engaging and accessible way to study anatomy, especially for students who require a little bit more time to understand certain anatomical concepts. They can, well, study it in their own time and in their preferred setting, which is great. So all in all, the models can form a great addition to students' repertoire of studying materials. But it's not just the students that can benefit from these models, because educators can use them during lectures, they could use them during assessment. And apart from their educational value, of course, they also safeguard the material. They safeguard the biological material because they don't really fall apart like what plastinated specimens would over time, slower than cadavers, of course, but still they go through the wear and tear. So, which is especially relevant for specimens with intricate details, for example, that would require a very or a long time to properly prepare them. And this way they can be well stored. They can even be in the future, maybe they could be 3D printed, so we could create a multitude of physical specimens based off of the original scans. So in conclusion, the digital model creation was very successful, and the models offer a very large array of possibilities. And we will still need a future study to properly assess them, but for now we're very, very positive. So this is the article that I mentioned as well. Feel free to scan the QR code. It will be visible in this slide and on the next slide as well, and also at the end of the presentation, if I'm not mistaken. So this project focused on anatomy learning, and anatomy is just the first step. So as veterinarians, we are trained to apply our anatomical knowledge to our patients so we can perform certain procedures, for example. And this not only requires anatomical knowledge, but it also requires knowledge of the procedures itself and lots of practice. So if we want to develop animal-free alternatives to trained professionals, such as veterinarians, we need to know how best to train them and which methods are effective. And for that, we need, of course, research and evidence, which Noemi will tell you all about. Thank you.
Thank you very much, Bart. We have now also a couple of minutes. If you have any question regarding this presentation from Bart, feel free to share it in the chat. And yes, if any question regarding the content, but also what we can do with it, feel free to share it before we move toward the next session with Noemi. So Bart, we have a question again from Nick. How was student feedback been on these models and techniques?
Yeah, so the feedback was, unfortunately, we haven't had any like very official feedback, but like the unofficial feedback that we gathered was very positive. Students really envisioned that they could use the 3D models, especially because they could use it just at home without needing to be well at the university to look at plastinated specimens, and not all of them are always available. So they seem very positive.
Thanks, Bart, and thanks, Nick, for your question. We have another question from Yonk. Do you see XR-based 3D anatomical models primarily as a replacement for the use of animals, or rather as a preparatory step before the after in-person experience?
Yeah, that's a great question. I think in the future, when all these techniques are way more developed, I think it would be great if extended reality based models could replace the use of animals. But right now it couldn't, and even then in the future, I think you still need these interactions with animal based materials. You need to have dissections to fully understand how tissues work, how the anatomy, well, what it looks like, how it feels. That's just something that, especially as a veterinarian, you need to experience. But it would, of course, help in kind of specializing those classes. So it would help in refining the classes that you do have with animal materials, I think.
Thanks, Bart, and thanks, Young, for your question. I let Noemi move to the next session. So yes, please feel free to go ahead.
You are muted, Noemi.
Yes, I realize it now. Sorry, can you hear me now?
Yes, we can hear you, yes.
Okay, perfect. Thank you for the feedback. So good evening, everyone. I am Noemi Passarello. I'm the postdoctoral researcher of the AvatarZoo research unit, and tonight I will talk about research. I will talk about all the research projects that we're working on currently and how we aim to translate these research outputs into evidence-based practices that we can then use for our educational settings. So I will start by saying that I sum up all our research projects into 3 main research lines. And the first one, I'm not going to go too much in depth about it because Bart literally just talked about it a few seconds ago. But it is just to say that this 3D digitalization of plastinate specimen, it is a nice of course, application of the AvatarZoo project as a learning and educational tool, but it is also a research project. As Bart said, we have recently published the publication for the methodology through which we achieve these digital plastinates, but we're still pretty much interested in understanding its usability. And as we mentioned already, how the students react to them and how can they benefit from these tools. Now, another research line that I will talk about is the 3D learning benefit research. Now, here I will talk about why learning in 3D is so important in terms of our cognitive function and our brain activity, and also how can this be achieved through extended reality and what learners can benefit from that the most. And a second research line that I will talk about has to do with the use of extended reality as a simulation for training the clinical procedure in a more safe, immersive, and repeatable learning environment. So before I do all of this, I want to take a small step back and talk again about something that Berend already mentioned in his introduction, which is the difference between learning from 2D stimuli and learning from 3D stimuli. Now, this is very important because this is the foundation and the main goal and hypothesis of all our research topic. Now, let's imagine that you acquire information from 2D materials, books, images, whatever is a flat type of information. You will store this information in a part of your brain that we call working memory. And you need to imagine working memory as a sort of an imaginary box where you just store all this information. Now, there is a lot of debate about this, but it's pretty much known that not a lot of people manipulate this information when they put them in the working memory box. They simply store them the way that they receive them. So in our case, it will be in a 2D format. Now, let's imagine that you need this information to perform some type of clinical procedure. You need to retrieve this information, so you need to take it out of this imaginary box. But then the situation is that this information will likely not be sufficient to do the clinical procedure because that 2D information doesn't give you the full spectrum and the full spatial understanding of for example, the animal anatomy that you have to know in order to perform the procedure. So there is an extra step that we need to do, and this step is a conversion in 3D. Let's imagine that, for example, we're going to put all these flat images of the same structure that we have seen from different angles. We need to put them all together and create a three-dimensional image. And this procedure, it's linked to some sort of effort. This is what we call cognitive effort or cognitive load. And the situation gets even more complicated because we know that this cognitive effort is very variable among individuals, because there are some people, some students that are naturally better at this. They have naturally better spatial understanding, whether because they have it innate or because the environment brought them to develop this competence more highly, but they are better than others. And so you can imagine that the learning outcome can be not the same for everyone, and learners who struggle with this conversion can be left behind. Of course, this problem will be solved if we acquire the information directly in the 3D format, because in that case, it is stored in 3D, we can retrieve it, and we don't need to do any conversion. The information is ready to use. And this will lead to less effort and, in theory, less individual differences. And I say in theory because, of course, who's naturally better at spatial understanding will always be quicker in learning. But at least the people that have struggled with this spatial understanding will struggle the least because they don't have to do the conversion. So, of course, we have said this already, learning from 3D, it is possible through traditional teaching methods, but unfortunately, this requires the use of animals or cadavers, and this is something that we cannot do so lightly anymore because we have ethical and practical concerns. So in all our research projects, or in the majority of our research projects, we try to use extended reality as a technology that can give us this full 3D interactive learning experience, but without relying on this type of traditional methods that can be challenging. So all our research activity started with the work that Berend has done already on human medicine. Berend has created some models of human anatomy, and he has tested among students to see how they react to it and if there is actually any differences among them in learning with different type of format. And Bo van Leuven, one of the PhD students from our research unit, has picked up on this research, and he has produced this output that we also managed to publish. And here he found that the format in which the information is delivered really impacts the spatial understanding of the students. What he found that the students who are lower in spatial understanding struggle the most with this 2.5 format, that is basically when we see a 3D information on a flat screen. And this happens because our brain doesn't really register a 3D image on a flat screen as 3D. We need a bit more immersion into the 3D space. So, of course, the next step of both research is basically to compare the 2D static learning situation with a full 3D interactive learning situation, both of which will be delivered through extended reality. And once again, the expectation is that the 3D interactive learning will cost less effort in general. And once again, we are interested in seeing also how individual differences plays a role into this outcome. Now, we have talked about this measure, the cognitive effort, but how do we measure it? How do we evaluate if there is any cognitive effort? Now, the most obvious thing is to ask the students how they felt during the task. Did they perceive the task as too much difficult? Did they found a lot of stress during the task? Did they perceive any cognitive load? And this is what we call the self-report questionnaires. This is a way of collecting data that is very popular because it's very easy, and we have also a lot of tools available to us because we have many questionnaires available. But this type of methodology has two very important limitations. The first one is that it is a retrospective measurement. So we're asking the student to remember his perception when he was doing the learning task. And the second difficulty is that it is based on perception. And of course, my perception of what is difficult is not the same perception of someone else. So this measurement can be very much biased. And one way to cope with this is to use brain activity. Now, I'm not going to go too much in details here, but just know that every type of action, every type of task that we do in our everyday life, including me talking right now and you listening to me, is related to a specific brain activity. And we can measure this brain activity through some technologies. For example, we use electroencephalography. And if we manage to measure the brain activity during for example, the conversion from 2D to 3D, we can have a more objective measurement of whether this effort is actually there and how big it is and how it varies among individuals. So this is a bit the idea behind the other research project that we're currently working on. For example, Rosamund Khan, another PhD student of our group, is working on finding exactly what are these neural markers that are associated with individual differences in spatial understanding and also in cognitive effort. And this is also something that I am personally working on in my own research project. And in this case, I'm replicating the situation that I've explained to you before about both experiment in which we compare a 2D static learning versus a 3D, more interactive learning situation. And then we ask students to judge spatial relationship between two structures. And during this task, which in theory, the students should do the conversion if they've learned in 2D or no conversion if they're learning in 3D, and we can measure their brain activity and find finally this objective marker of cognitive effort. And once again, of course, the picture is not complete if we don't take into account also the individual differences, so how naturally people are good or bad in spatial understanding. Now, another research line that I will briefly mention has to do with the use of extended reality as a stimulation for training clinical procedures. Now, so far, I've talked about how we use this tool to study, for example, static anatomy to judge spatial understanding between structure, but we can also use the extended reality and the 3D learning environment to train specific clinical procedure where the 3D learning is very important. So, for example, Nelleke Venema, which is another PhD student from our group, she is working on this project that already Berend mentioned briefly before, in which the student can interact with this cow model. They can lift the tail up and they can perform this injection. So the idea behind this project is not just to study how well the students learn, so how good is their performance, how it improves over time, but it's also to study how the perception of the procedure change over repeated exposure. Because you can imagine that performing this procedure on a real animal for the first time can be quite demanding in terms of stress, anxiety. So of course, practicing this procedure, at least from a mechanical point of view, we hope that can boost the students' self-efficacy and confidence. So Nelleke is working on this and collecting some data on this model. Something very similar that we're working on, specifically me and Bart are working on this, is our dog intra-articular injection model, in which we train another clinical procedure. Now here we're dealing with osteoarthritis, which is a very common pathology amongst both animals and humans. And we know that both for the diagnosis and the treatment of this pathology, we need to perform a very specific type of injection. And this procedure is currently undertrained, unfortunately, in our educational curricula. And the reason why this is undertrained is, of course, because it requires the use of animals or cadavers. And we have said already that we cannot do this lightly anymore. So our solution is to create this XR version of the knee joint, both for dogs and also for humans. And we want to use this model to train both students and professionals. At the moment, we are in the phase 1 of this project, in which we are still developing the model. And then we will go in phase 2, in which we validate the models and how it works, same as we did for the cow model and all our other AvatarZoo models. Now, I want to close by talking briefly about what are our future milestones in terms of research, because all the projects that I described to you, they are still very much open projects, but we are already aiming to future projects. First of all, we want to go a bit deeper into this neural dynamics of the 3D learning experience. So far, we are focusing so much on what happened after we have learned, so when we need to retrieve the information, but it will be also interesting to see what happened in our brain when we're learning in 3D compared to 2D. Something also that we're interested in, because our technology thankfully allow us to do so, is to find other outcomes that can objectively measure cognitive effort. For example, eye tracking. We know that some eye movement measurement or pupil dilatation or eye fixation, they can be connected to attention and to cognitive load. So we're also very much interested in looking into this. And something that also we want to include in our models are extra features. For example, also this was mentioned already, we are working on some sort of integration of the haptic feedback. At the moment, we are only talked about this in theory, but of course, this is one of the main critique that you always encounter when you talk about extended reality as an educational tool, because of course extended reality technology mostly use visual information to train participants, and sometimes for our brain visual information is not enough to actually learn something. So I went very quick into all our research projects because we have so many open, but if you're curious about anything, please feel free to ask me something now, or to scan our QR code, or communicate with us through our email. And thank you so much for your attention.
Thank you very much, Noemi. Right on time. I think we still have time for one or two questions, and after for the closure. I see one question in the chat already, Noemi, for you. Is less cognitive effort always a good thing? Does it depend on what is being studied, what skill is being practiced? This is a question from Nick.
This depends on how we think about cognitive effort. So in this context, the cognitive effort is really the fatigue that we do to translate this type of information. So of course, in a different context, for example, struggling a little bit with an information can also help understanding the information a little bit better, right? The repetition can also be a measure of learning. But in this context, when we talk about procedural training, so when we need to learn to perform something, this struggle can make the response slower. So if we have to react to a stimuli, we will be slower, we will be less accurate. So in this scenario, it's not always ideal.
Thanks, Noemi. And one last question for any of the speakers from Liliana. What budget would be necessary, and how long will it approximately take if a university without any prior experience in XR models would like to introduce a type of AvatarZoo?
Okay, this is an interesting question.
Maybe can I
yes,
that
go
I
ahead.
can
In 1
answer
minute.
that. In
Yeah.
1 minute, okay. So it is a it will be a large investment. I hope that by sharing the AvatarZoo in the future, we will be able to help you with that. So speaking for myself, I've been busy close to 10 years now with this technology and the research behind it. And of course, when you want to introduce it, just introduce it in your education, because you have seen that our research shows that it really works, that it will take you a lot less time. But it will be a major investment to do all the models, and because the models that are out there are not sufficient for our type of education, so they need to be adapted, and then you need the platform. So we hope that in a couple of months, I hope in a couple of months, we will be able to share the first models with you in the context of the AvatarZoo, and then you just, between brackets, need the hardware to show it to your students. I hope that answers your question a bit.
Thank you, Beren. Yes, please, Daniela.
Yeah, I wanted to share with the audience that in the Netherlands, for in the human field, more grants have been given for developing XR tools than for the veterinary field. At the moment, there are large initiatives for virtual reality, to bring virtual reality in the operation room, to bring virtual reality for training of nurses and for communication with patients. So we are trying to keep up with what is happening in the human field, and yeah, it would be really nice to be able also to create perhaps a coalition of life sciences or veterinary faculty that could work together, developing and sharing these models. So if somebody in the audience feels to connect, I think via Valentine or one of us, please connect with us.
And maybe I can add to that a little bit. What Daniela said is true, and I hope you appreciate that the research we are doing essentially doesn't have to do a lot with the veterinary field, but it is all about learning, getting spatial knowledge from 3 or 2D resources. So that's a very generic concept, and you can apply that to human medicine, but also to many other parts of education. So the educational concept is very, very, very generic.
Thank you, Berend. I think that we are reaching the end of this webinar. Thank you for staying with us and for listening to this very nice and complex work. I think that now is the time to announce the next webinar. Somehow I did at the beginning, but I should not have done it. So, Nick, please go ahead.
Thanks. So yes, just another reminder, the information has gone into the chat. The next webinar being next February the 18th. So you'll be able to read about that and get all the information. I'll just check the time again, 10 a.m. CET, Central European Time. And Brett Lidbury from the Australian National University will be talking about alternatives and simulation in education as part of this webinar series. So do join us for that. Looking forward to seeing some people again. This series is the Alternatives and Simulation in Education webinar series. This has been the third webinar, and this is a series that was developed following on from the International Conference on Alternatives and Simulation in Education held at the end of August last year as a satellite conference to the 13th World Congress on Alternatives and Animal Use in the Life Sciences. So these will be monthly webinars, and thank you everyone today. Look forward to seeing you at future events, and I'll pass back to our host, Daniela. And
thank you.
Valentin.
Yes, first of all, I would like to thank you everybody again, but also Valentine for organizing this evening. Thank you very much. I would like to thank Noemi, Bart and Berend, the presenter of this evening. I would like also to ask the audience that if there are topics that they would like to hear about during the next coming months, Katrin, Nick and I are trying to make prepare at least a 6-month program so that we can present in advance. So we will do it during the coming weeks and share it with you. And if you would like to connect with us for suggestion, for some advices to how to proceed and topics, please connect with us. And again, thank you very much and have a very good evening.
Bye, everyone. Good evening, and thanks for your participation.
Good evening. Bye, everyone.
Bye-bye.